Every day, we are losing more and more of our planet's wildlife. But did you know that by biobanking cells, we can preserve the biological material that's essential for life and actually help to save animals from extinction? Nature Safe is pioneering a new era in conservation by doing just that. So far, we have preserved over 300 species. From land to sea, nature needs us. Help us to keep nature safe with a secure donation today.
Hello, and welcome back to Restore and Pet Podcast with me, your host, Jack Cole. So today I'm joined by Debbie Rolmanis from Nature Safe. So Debbie, welcome. Thank you so much for your time, and start by telling us a little bit about your background and the work it is that you do over at Nature Safe.
Well, hi, Jack. Thank you so much for having me on the podcast. I'm really excited to be here. So, I'm Debbie Rolmanis, and I've been working with Nature Safe since it started in 2020. My background isn't particularly conservation orientated, so I'm coming a little bit out of left field into this work. So I started life as a high performance sport biomechanist and coach, and then moved into a bit of business development and business consultancy. It's always really been about growing things in a positive direction. So whether that's an athlete or a business, that has been sort of my background, and making things a bit better than they were before. So then I met Tullis Matson, who is the founder of Nature Safe. We worked together on a committee, and he was talking about the charity that he wanted to set up, which was Nature Safe. And I said, that sounds amazing. I've always been a nature lover and involved in the countryside. And so I said, how can I help? So I came on board initially as a volunteer to get things going, doing some social media, bit of branding, that kind of thing for the charity, and then came onto the board as a trustee, was on the board for a couple of years, and then last August I moved into the role of COO in a full-time role. So we're a very small team where I'm the only full-time paid member of staff, and we've got a couple of other part-time members of staff running it all. So that's how I got here. And every day is a learning day for me, which I absolutely love. But for me, it's all about making life better, preserving the life we have, and growing this charity to be able to have greater impact on our conservation and biodiversity. So
Brilliant. So tell us exactly what it is that Nature Safe is doing in saving animals.
Sure. So Nature Safe is a charitable living biobank. So we preserve cells from endangered species across the whole remit, from least vulnerable to critically endangered and extinct in the wild species in a living state. So we freeze samples at -196 in liquid nitrogen in a way that ensures their viability for the future. So we collect gametes, so reproductive samples, and skin samples from species that come into us, and as I say, freeze them in a way that means that they will be able to be used in assisted reproductive methodologies in the future.
Brilliant. And give us some examples of some of the species that you've got there.
Wow, we have 278 species safely stored, which in 4 years, I think that's pretty good going. Actually, we're really we're in a race against time for a number of species, and so that's why the count of those species is so important. It's not that we want to make sure that we're banking as many as we can. So we've got everything from UK natives, so red squirrels, hedgehogs, Scottish wildcat, up to black rhino, elephant, lion, and everything in between. We've got amphibians, birds, fish, and the list. Yeah, we've got a multitude of. We've got all taxa in the tank, and from all pockets of the globe, basically. So.
Brilliant. And so what exactly are the incentives for conserving these species into the future?
Sure. So, as you know, our biodiversity has been rapidly declining. Over 46,000 species are at risk of extinction, and human activity is the main culprit for this acceleration of extinction in our wild world. So, the incentive is to bank this genetic diversity that we've currently got, because what happens in an extinction, there are a number of reasons that populations go extinct, but one being they enter into this extinction vortex, which is when the population numbers start to dwindle so much that inbreeding occurs, and of course inbreeding results in animals that are not viable for long-term health, and so that tends to deplete the population even more. So what's critically important is that we are preserving genetic diversity to avoid that bottleneck of inbreeding. So, and that, as things decline, you can understand that as that's what I was saying about the race against time, it's incredibly important that we bank what we currently have, because once it's gone forever. And there are a number of reasons for us to be doing this biobanking work. So there are a number. We work with conservation organisations throughout the UK, and they will largely be involved in breeding programs for specific species or a subgroup of species, and they manage those populations in captivity incredibly well. So what we can do is preserve the genetic diversity of captive populations, and quite often they will rely on working with other organisations, bringing animals into a collection to ensure that it's genetically diverse to what they've already got. So there's a lot of animal movement going on, which sometimes can be really challenging and can take quite a long time. Some animals might not be fertile, or you might lose one half of the breeding pair, and so your opportunities for reproducing and continuing that species starts to dwindle. So what we do is, by being able to preserve those samples, we provide an opportunity to reproduce long after an animal may have passed away with their genetics, or even if moving animals becomes a little bit more challenging, potentially this provides the opportunity to use these samples without having to actually physically get an animal in place. So for captive breeding, that's sort of some of the incentives around biobanking. But in situ scenarios, so our broader mission is to try and develop these living biobank hubs in geographic locations around the world. So strategic places that biodiversity hotspots, or at least places where some of these animals you can only find in those regions. It's so important that they have the capacity and the ability to preserve these samples on site, basically in country, because with all the restrictions about moving samples around the world, then it becomes incredibly challenging to often get the number of samples that you need to make sure a population is viable, which is, we go on the basis of 50 genetically diverse samples. So by enabling conservation organisations around the world to have their own living biobank, it means that wild populations can be supported as well. So yeah, it's preserving the genetic diversity of what we currently have in a way that is supportive of the species and ensures that we have options, really, to make sure that we don't lose any more. And of course, we always say we are one tool in the conservation toolbox. Our house needs to be in order in terms of habitat and what all human activity, that what we're doing to the planet. But whilst we're trying to fix those other things, and people are doing incredible work, other organisations doing incredible work, yourselves included, to try and to fix what else is going wrong, at least we are safeguarding our wildlife. so that we have, as I say, we have options and a bit of hope for the future, that should we hopefully get our habitats in order, that we have some animals that can actually enjoy living there again.
Brilliant. And how does it work? To take the gametes, the genes, the stored from the biobank, and then how do you say, not reproduce it, but then how do you I don't know, quite sure what the right word is, but how do you then birth the new species from the genetic material?
So because we can store sperm and eggs in a living state. So you can cryopreserve sperm and then thaw it, and it can be inseminated as they would for domestic animal breeding purposes. You can apply the same techniques into wild species. We are also the reproductive samples can be quite challenging because we do need them within a short time frame to ensure their viability. But we also take skin samples, which they have a bit more of a longer, so they've got 5 days for organisations to get those skin samples to us, and they're still viable within those 5 days. So that's a bit more interesting in terms of the science and what could potentially be done using the skin samples. But they have in mice now been able to switch a skin sample into a sperm or an egg, and which means that, and of course skin regenerates rapidly. You cut yourself, you grow a scab, that's just skin cells proliferating really, really rapidly. And we've grown cheetah cells, we've grown a couple of others, I can't think of the top of my head, but we've grown a couple of cell lines up because we can regenerate skin cells, and they contain the entire DNA of that animal. So, and they're alive. And so that as science and tech develop, we have the opportunity to use those skin cells to then generate a reproductive organism, which can then be used in assisted reproductive technologies. Yeah.
Mind-blowing.
I know.
All right. So tell us a little bit about your day-to-day work, and it'd be a nice opportunity to hear about some of the butterfly work that you've currently got going on.
Absolutely. So cryopreservation is a really interesting one, and particularly for preserving things in a living state. There's an awful lot of taxa that have never been cryopreserved successfully before. So our day-to-day work is we receive samples we've got 50 UK conservation organisations as partners. So they are continually sending samples in to us for processing. We wanted to remove all the barriers to conservation capability for our partners. So we run a free service. So samples come in to the lab in Whitchurch in Shropshire, and our lab team will process those samples. Alongside that, we are always developing protocols. So, for example, amphibians are notoriously quite challenging to successfully cryopreserve. So we're working with, got a fantastic science committee of global scientists that help us develop protocols for that, and like some seahorses and all sorts of things. And the one that's coming online in the next couple of years is butterfly larvae. So, as you know, butterflies are a massively important pollinator, and many of our UK species of butterfly have declined hugely. So we are partnering with Anglia Ruskin University and Jimmy Doherty from Jimmy's Farm to run a PhD program to develop cryopreservation protocols for butterfly larvae, which is hugely exciting. And then it all coincides with habitat recreation and wilding of areas, so that we have somewhere to release these butterflies back into. So that's an example of one of the projects that we're pushing forward. The other one that we are really excited about, and just before Christmas we had some huge success with, is coral cryopreservation. So we are part of the Cryo Corals network that's been recently established in the UK, with a number of organisations involved in that group. including London Zoo and Edinburgh Zoo and the Horniman Museum and ourselves. And we, just before Christmas, Tullis and I went down to the Horniman Museum because they were having a spawning event, which, for somebody that hasn't come from the conservation world, was absolutely incredible. I can't tell you how magical it was. And so an Australian species of coral spawned, and we had taken the day before to get all the freezing curves and all the protocols sorted for the coral sperm. And then we froze the coral sperm that day. And the next day it was thawed and a successful embryo fertilization occurred. So we actually made baby coral from sperm that had been frozen the day before. So hugely successful. And for me, that just provides a wealth of opportunity in the coral space. 40% of corals are at risk of extinction. We have to act now. We have to be able to do something. And for some coral species, biobanking them is their only chance of survival. And so being able to provide this opportunity for the other conservation scientists who are doing all this research around how to potentially support the breeding of corals so they're more resilient to climate change, all of these things, they now have this opportunity to use some of this frozen sperm to conduct some of these experiments, whereas before the time frame was so tiny that they potentially didn't have a chance to do it. So it's those things that are just so exciting about what can be done. And it's thanks that protocol came from Mary Hogdom in the US. I think she works with the Smithsonian, and so she's sort of coral guru. So we implemented her protocol for that. But again, it's this idea of collaboration and network and doing things for the greater good and sharing expertise that we're all about. So yeah, so that was very exciting. But and again, I think I've mentioned seahorses before, but that's another area that I don't believe has been done before. And so that's an exciting one that we're tapping into as well.
And these samples, just out of curiosity, because often we think about, sci-fi and sort of people freezing themselves after they die and this sort of thing. Is there a time frame on a sample once it's been frozen?
No, it's basically indefinitely. So yes, so what happens, we dehydrate the cell when we're processing it and then basically put antifreeze in it. That's, in layman's terms, that's what happens. But that antifreeze needs to be different for each species. But the idea is they are suspended in animation for as long as you need them to be. And so, yes, it technically is indefinitely.
Goodness. Okay, and tell us a little bit about how this field has progressed, sort of, if you can tell us how perhaps it started and when things got going and where things could end up in 5 to 10 years, how the technology is improving.
Sure. So I think San Diego Zoo were probably the first ones to start this sort of living cryopreservation techniques, and that was about 40 years ago. There haven't been many living biobanks set up from that. We are the only charitable one in the UK and Europe. There are many biobanks around, but biobanks that store DNA only, and there's the cells and the DNA are not in a living state, but you can use that DNA for genomic research and all heaps of valuable things, but it can't be used for regenerating life. So for us, I think sort of in terms of mammalian cryopreservation, we've got that fairly sorted in terms of protocol and process. For us, it's the development of these protocols across different taxa, and that is where I think this constant research and science is happening. For me, I'm not the scientist, but I know that science works at a rapid pace. It really does accelerate. There are some incredibly smart people doing incredible things in science. And so there are so many tools at our disposal now to as long as we've got the sample, that's the key thing. As long as we've got the sample, there are so many tools that we can draw upon to make sure that we can keep some species alive. Where it's going, gosh, you know, that's a massive question, really, and it feels almost that the world's your oyster. It's interesting to talk about. We're not about de-extinction at all. You know, we're about preserving what we currently have and making sure that the populations that we have can be robust and vital. But colossal in America with the woolly mammoth and all that kind of thing. Whether you like it or not, and the question that I always ask is, just because we can, does that mean we should? But that aside, the technology that they might be developing from going through that process may be really interesting from a conservation standpoint. So I think it's important that we maintain a healthy level of open-mindedness and curiosity around what developments are happening in science, but also to maintain an equally healthy idea of, you know, how much should we be doing in terms of this science and this tech. But it's interesting, and you know, and I think we definitely there's an argument to stay curious, because an awful lot of good stuff can happen.
Absolutely. Wow. Yeah, and finally, Debbie, where can people find you? Nature's Safe, to donate, support your work, or otherwise get involved?
Yeah, well, we are a charity, so we rely on individual donors and grants or trusts or corporates. So we can't do any of this without funding. And I know there are so many causes that everybody can donate to. But yes, this is our little plea for help. So we at our website www.natures-safe.com. We are on Facebook and Twitter. No, we're not. We're on Facebook, Instagram, and LinkedIn. Nature's Safe for all of those. And yeah, you know, find us there. And any donation is just incredible. We are focusing as well on UK native species. So, you know, anything. We've got all sorts going on. So hedgehogs, red squirrels, you name it. And we want to expand what we can protect on our UK shores as well, because we are one of the least biodiverse countries in the world, as I understand, which is terrifying. So we are doing awful lot of work with the UK natives, as well as all of the other animals that we've got. So any help is gratefully received, or if anybody has any contacts or anything that they would want to do or fundraise for us, then you know we would love to hear from you. So yeah,
Brilliant, Debbie. Thank you so much for your time.
well, thank you, Jack. Thank you for having me. That's
More than
great.
welcome.